Seems I'm no longer presenting. Hello, can you just reply to the chat room if you can still see my slide? Okay, amazing, amazing. Thank you guys. Thank you guys. Amazing, amazing, amazing. Thank you. So I'm going to be sharing my slide. I'm going to be sharing my slide once again. Thank you guys. Thank you so much. Thank you so much for replying. So let's see. Let me share my screen again. So can you see my slide now? Hello? Can you see my slide right now? Yes. Yeah, we're recording. Hello, can you see my screen? I hope my screen can be seen. Can you? Okay, thank you, thank you. So the use of bioinformatics pipeline tools and database for omics data analysis. So this is the workshop. And after my session, we're going to welcome Dr. Shabana Bibi to take our aspect on pharmacophore and bioinformatics. Now, let me proceed. The use of bioinformatics pipeline tools. Now, what is bioinformatics? Let me start from there. Bioinformatics is is biology doing stuff on computer, or is the process of using the computer, all right, to analyze biological data or biomolecular data. All right, that's bioinformatics. And in this bioinformatics, it's actually subdivided into different aspects, because in bioinformatics we have the genomics, in bioinformatics we have the transcriptomics, in bioinformatics we have the proteomics, in bioinformatics we have the metabolomics, in bioinformatics we have the metagenomics, in bioinformatics we have the pathogenomics. In bioinformatics, we have clinical genomics. In bioinformatics, we also have precision application of bioinformatics in precision medicine. We have the application of bioinformatics in agriculture. So I'm going to show you all these things, all right? So, but you see, you cannot call yourself a bioinformatician, or you cannot actually be relevant or be able to actually explore bioinformatics without actually using pipeline tools, all right? You cannot actually explore this area without actually using pipeline tools. Now, before I proceed, actually bioinformatics is actually a lucrative field of study, and is an interdisciplinary area of study. All right, bioinformatics is an interdisciplinary area of study. It cuts across different life sciences. It has applications in different life sciences, either in the health sector, in the agricultural sector, in wherever. And it has application in the academic sector. It has applications in the industrial sector. All right. It has application in the research space and the biotech space, as far as all this life science or biological related field is concerned. All right. So, and then you see the career prospect about bioinformatics. We're also going to be talking about next month. Okay, the career prospect in bioinformatics, the present market value of bioinformatics, the salary of bioinformatics. All right, especially in the US, a bioinformatician is actually being paid $125,000. All right, per annum. All right, $125,000 per annum. bioinformaticians. All right, and then we have the marketplace of bioinformatics, where it deals with freelancers who analyze omics data for clients. All right, we have the aspect of bioinformatics in terms of capacity building. We have platforms, we have companies, we have organizations even like us that we offer bioinformatics training and capacity building, research training, research mentorship, research guidance. Now, these are the marketplace of bioinformatics. These are the areas of bioinformatics in the marketplace. All right, so what I'm just trying to say in essence is this: bioinformatics cut across different fields, and we have as far now, we have what we call pipeline tools. So if you want to explore bioinformatics, or you want to have skills in bioinformatics, it is that you need to be able to know how to use pipeline tools and databases for omics data analysis. Now, there are two aspects of bioinformatics, all right? Or there are two aspects as far as biology is concerned in computer. The first one is computational biology, and the second one is bioinformatician, all right? So just like in data, all right, in data space, we have the data scientist and the data analyst, all right? The data scientist and data analyst. Now, in biology relating to computer, we have data scientists who are computational biologists that build pipeline tools, that build softwares, all right, that build softwares for to actually use this software for omics data analysis and come up with tangible insights, informations, relevant informations, predicted genes, all right, informations about genes that are responsible for disease, genes that are responsible for an expression of a particular phenotype in an organism, all right, genes that are responsible for an outbreak, genes that are responsible for a pandemic or whatever plague it is, right? So because when you use these pipeline tools that are developed by computational biologists, as a bioinformatician, you'll be able to use these tools to do omics data analysis. Now, when we talk about omics, we are talking about genomics. We are talking about genomics when it comes to DNA, using DNA samples, okay, or DNA sequences from organisms that you can use it to actually use these tools to actually analyze this DNA, because this DNA is the coding information, all right, of the genetic information of certain functional properties or features of an organism. Do you understand? Because that is DNA. DNA is a functional hereditary material of an organism. So when you get the DNA, which is actually the source of every functional structural component of an organism, by the time you get those DNA, you actually be able to use it to do quite a number of different kind of analysis, come up with workflow, bioinformatics workflow, and then you can actually use it to analyze those data and come up with an insight, information that is relevant for research paper, information that is relevant for research, information that is relevant to develop new drugs, information that is relevant to come up with new digital software tools that can actually identify biological biomarkers that is specific to a particular disease, phenotype or genotype or whatever it may be, okay? Or even for diagnosis. So we are going to actually explore some of this area. And I'm going to show you some practical applications of these bioinformatics software tools and pipeline applications to do quite a number of different kind of analysis. Some people are saying they cannot hear my voice. So please, can you reply at the chat room? Can you hear me clearly? Can you hear me clearly, and can you see the slide? Okay, I'm going to share the slide. Is my voice clear, and can you see the slide? Can you see the slide now? I believe you should be able to, right? Okay. Okay, so thank you so much. It seems you can actually see my slide. Okay. Thank you so much. Thank you so much. Okay, thank you guys. Thank you guys. Amazing, 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 amazing. Okay, so let's continue. Like I was saying, so the use of bioinformatics pipeline tools and database for omics data analysis. So I am trying to say in essence that if you want to explore this area or field of study which is lucrative, which is trending in the life science space, right, you need to be able to know how to use pipeline tools. And I was explaining something to you. I said computational biologists actually uses biological data, all right, to analyze what? Biological data. Now, the next thing I said was that you can actually use this biological data as a bioinformatician, now not as computational biologist. All right, as a bioinformatician, which is like a data analyst, to actually explore omics data analysis for omics data for for doing genomics analysis or transcriptomics analysis. Now, I was explaining genomics, right? That genomics deals with DNA. So when you get a DNA sample or DNA sequence of an organism, you'll be able to use this pipeline tools or database to analyze those nucleotide sequences and come up with an insight that can actually be used for further research, like drug development, like to understand the outbreak of disease, to understand what is responsible and what is not responsible, to understand genes that are expressing and genes that are not expressing, that are dormant or that are knocked down. You get the point now. So that is so bioinformatics is interesting. It's interesting and it's very, very interesting. And industries like biotech industries are actually looking for bioinformatics, especially in Africa. In Africa, bioinformatics is gaining ground. Genomics is gaining ground. It's gaining ground already. Now you can hear a lot of news out there, right, about so many things that are actually going on as far as bioinformatics is concerned. And some of these things that are going on is that there are startup companies, big tech companies, genomic companies that are coming up, with sequencing platforms, machines, some big tech companies that can actually analyze big data, during COVID-19, and before they could actually understand the phenotype and morphological features of COVID, they actually used bioinformatics to explore these things. And then they now began to see, oh, these are some of the genes that are present. This is the gene, and this is, and there was a time that, COVID-19 began to, become more virulent, it was mutating. And what is the last, can somebody reply in the chat room? Okay, what was the last name of COVID? Mutated COVID. What was the last emergence of a new mutation of COVID? Somebody can reply quickly. What was it? Yeah, somebody just got it. Who else can give us? Is there anything more apart from that? Thank you, guys. Thank you. You all are correct. Omicron. You all are correct. Omicron. Now, the reason why we add Omicron as a COVID, right? As COVID was because of the emergence of a new kind of a mutation that occurred. All right, a new kind of mutation that occurred that changed the morphological behavior or the virulent behavior, the pathogenic behavior of COVID. You understand? It was because COVID kept mutating, and once it mutated to a different kind of COVID because of the genomic structure. All right, because of the DNA sequential structure pattern of COVID that is different from the earlier COVID that actually break out from China. And that is why you have to map it to reference. So they did some kind of mapping to reference, and they mapped the Omicron to the reference. That is where they could actually say, oh, it is an Omicron, because there has been an evolution over time. There has been an evolutionary change over time. Now, this is bioinformatics for you, because they could actually discover that this is Omicron because they use bioinformatics sorry, the chat box, we are not being able to do that. We'll do that later. Okay, we'll do that later. Okay, thank you. So what do you imagine as a life scientist? So as a life scientist, you must be able to actually get to understand these things. And by the time you do, you'll be able to actually use these tools, all right, this understanding, these skills, this capacity to do a quite number of different research based on what you imagine as a life scientist, okay? Based on what you imagine as a life scientist. Now, what did I say about that? Or what did I mean about that? Okay. You as a life scientist, you have a vision, you have a call, you have what you want to do. You bet probably your call, in your vision, your goal is that you want to resolve cancer. Your goal is that you want to resolve a specific disease, or your goal is that you want to do a groundbreaking discovery. Okay, and before you can do this, actually, you need capacity. You need to be able to gain capacity. You need to be able to gain skills, to be able to understand the. And then I'm going to take you to different aspects of mass, okay, in pipeline tools and applications. So don't forget to pipeline. We have pipeline tools on GitHub, all right, that are coded, and then we have some pipeline tools that are also coded, but they are online pipeline tools. Okay, so we have some pipeline tools that you need maybe Python or some certain aspect of computational software tools to be able to run those kind of pipeline tools for analysis. And then we have pipeline tools for database, on databases, which is online pipeline tools on those databases for different omics data analysis. Now, you see, one thing about science is that you keep discovering new things and making things that are hard over time to be okay. Okay, so we have different research areas that you can. I said you can actually use bioinformatics for. So we have pathogenomics. Okay. So we have the pathogenomics, which is study encoded microbe violence microorganisms to infect a host and cause disease. And then we have the probiotic genomics, and then we have the metagenomics. This was just an area of bioinformatics that you can actually use pipeline tools for different kind of analysis. Then we have the outbreak detection and surveillance. We have the diagnosis. We have some medicine. We have the drug discovery. We have the nutrition and diet. All right. Then we have the applications to agriculture. Once again, can you hear me clearly? Somebody just said the voice is not. It's not clear enough. Okay. Can you hear me clearly now? Is it clear? Probably it's your network. Yeah, I just dropped the slide. I'm going to be sharing something else. Okay, I'm going to be sharing something else from the browser. So I just want to introduce you guys to different pipeline tools and applications, all right? Different pipeline tools and applications for data analysis. Different pipeline tools and applications for data analysis. And then I'm going to practice a particular data analysis with you guys, okay? Yeah, we just dropped the slide. Please, can you hear me clearly now? I'm going to be sharing my screen once again. I'm going to be sharing. So please give us a minute. Yeah, hello everybody. So I hope you can hear me. Sorry, we had to a bit lock the screen. Lock the WhatsApp group for certain specific purposes. So I'm going to be sharing with you. Okay, I'm going to be sharing with you. My screen. All right, from the other end of the laptop. Okay. Hello. I hope you can see my screen. Can you see my screen? Hello. I hope you can see my screen. Sorry. So unlock it. So I'm going to be unlocking the group again. Can you see the screen? I need a yes at the WhatsApp. Okay, thank you. Thank you. So now I proceed. All right, so let's proceed. Now, this is Genius software. Okay, what you can see on your screen right now is Genius software. Genius 9.0.5 for non-commercial use only. So, but this particular software has a lot of plugins that serve like a pipeline tools. All right. So it has a lot of what? Plugins. And these plugins are used to analyze different aspects of bioinformatics, data. or different types of omics data. So we have different pipeline tools for bioinformatics analysis, okay? To analyze different kinds of omics data. So including those of genomics, those of transcriptomics, those of metagenomics, okay? Those of so in different aspects of genomics, okay? This particular bioinformatics tool, sorry for that. I think I was a bit distracted here. So, like I said, this is Genius software, and this Genius software is actually sold. All right, it's sold, but we have the activation key. All right, we have the activation key that you can actually use to use this this software. This software for different kinds of bioinformatics analysis. Okay, so and I'm going to take you through this software, and you can have free trial. You can actually use free trial with this software. So at the end of this meeting, we are going to send you a link that you can use to actually download this version of the software. Okay, we're going to send you a link that you can actually use to analyze data using this version of the software. Okay, so now this software can like I said, specifically it can do phylogenomics analysis. This particular software can actually train genomic data. This particular software can also does transcriptome data analysis. This particular software can do variant calling. This particular software, you can actually use it to also do genome annotation. You can use this software to retrieve data from the public access database. So the first thing I'm going to be introducing you is number one, the software, which is this. Then the next thing I'm going to introduce you to is online pipeline tools for different kinds of omics data analysis. Now, this is Genius software, and I'm going to provide you with the link, like I said. So when you open this software, all right, go to file, and from this file, you can actually import data. You can use the software from the file space to import data from your computer. All right. So, and this data you import, you can then use different tools on this particular software for data analysis. So, right here, you can see different folders. These are folders that contain nucleotide sequences and samples. Okay. And from here, you can have edit, which is edit option to just edit some documents, to copy some things. And then here we have the view. All right, here we have the tools. So there are some tools here you can actually use, for different kinds of bioinformatics analysis. And then here we have the sequence. And here with this particular option, you can have some options that you can actually work on sequences. You can have some options here to do certain annotation and predictions as well. Okay. Don't forget, this is Genius software version 9.0.5. In case you also want to download it from Google, okay? You can actually do that as well. So let's proceed. Now, the first thing I'm going to show you is how to import data from the computer. So, but to do that, click on local and create a new folder. Click on local and create a new folder. And this folder that you created, you can actually give it any name you want. Okay? You can actually give it any name you want. Okay? So let's give it example. Okay? So can you see the folder? So just click it and go to file, and then go to import. Then you import from file. All right, you import from file. So now, as you can see, these are data. These are DNA data samples. And these data samples are ITS sample, interspace transcript spacer sample, or Sanger sequence sample. Now, you can click two of it because this sample you see, one is forward and the other is reverse. So this is two samples belonging to one organism, or two data files belonging to a sample. So just upload it. This is a kind of analysis that you do for DNA sequence, okay? DNA sequence. And this DNA sequence, like I said, is Sanger sequence. Now, you can actually get Sanger sequence maybe for your studies, okay? Or you do Sanger sequence maybe for, you get Sanger sequence maybe from a PhD, all right? From a PhD holder. So by the time you do that, actually you'll be able to use it, okay? You'll be able to use this particular software for analyzing this kind of data. So this is ITS sequence, like I said. Now, the next thing you have to do is, okay, double click the sample. Remember I told you these two samples, these two files belongs to a sample, okay? I said these two files belong to a sample, okay? These two files belong to a sample. Please, can you see my screen very well? Can you see my screen very well? Can you reply to the chat room? Is it clear? At the WhatsApp chat room, okay? WhatsApp group. Amazing. Thank you so much. Thank you so much. Thank you. Thank you for your reply. So let's proceed. Let's proceed. So we have two samples. Now, from this sample, you can actually align them, okay? So you can do pairwise alignment. So that's the first thing you need to do. You need to do pairwise alignment, okay? You need to do pairwise alignment. And the moment you do that, you click OK, all right? So you can actually practice this when you get the sample. So you use muzzle alignment. That the moment you're done with muzzle alignment, click this home icon and zoom. Now, by the time you do that, you will see certain different kinds of things. Now, this is what I'll be teaching you mainly. So you can actually edit this sample, because when the sample came from the sequencing platform, it came with some errors, some alterations, okay? And that is why you can actually, that is why you need this particular software to be able to align it and edit this sample. So you can allow editing here. And the moment you do that, you can actually edit this, these errors, these alterations you are seeing here. It's not actually an error, it's just that information wasn't filled, okay? So you need to do that manually as far as Sanger sequence is concerned. This Sanger sequence is 16S RNA. I repeat myself, this sequence is 16S rRNA sample, okay? 16S rRNA sample. Now, by the time you are done, okay, by the time you are done, you try to edit it, okay? You try to edit it. You click allow editing and you click backspace. Then you click OK, then you'll be able to allow editing. Now remember, we add K there. Now when you place your cursor, all right, your cursor on the K and look down here, okay? Look down here. So let me zoom. When you look down here, so let me place my cursor on K, all right? I hope you can see my screen clearly. Hello, can you see my screen clearly? You can't reply. I hope you're following. Thank you, thank you, thank you. So let's proceed. When you place your cursor on K, you will see something like mouse over column. K is either G or T. Quality 1, right? Minus quality 1. So it means K is either G or T. Now remember, in DNA we have ACTG. The A is adenine, guanine, cytosine, and thymine, right? A, adenine, G, guanine, C, cytosine, and T, thymine. So, and that is what make up the entire genetic structure of an organism or a particular gene, okay? Sequence pattern, all right? So when you click this K, when you click here, right? Click right in the front of K, click backspace, you'll be able to delete it. Okay, so let me do Control Z to return it. So remember, we place a cursor on K, it tells us it's either G or T. So it means this K is either G or T. But you have to edit this sequence using the what's it called? Using the chromatogram graph. This graph you see, can you see different colored graph? You can see the A, the C, the G, and the T, right? So the A is red from this end, the A is red, the C, the G, and the T. All right? The A, the C, the G, and the T. I hope you're following. Hello? Please, are you following? You can reply the chat room if you are. You can reply with a yes at the chat room if you are following. So let's proceed. So we have, so you have to click backspace, K, because here we have G. Now, what is the highest graph here? Green is the highest expressed graph, and green is T, right? The green is T. So the K here would be T. And when you place your cursor on K, it's telling you K is either G or T. So what's supposed to be here is not G, but what? T. So you click backspace and you impute T. As you can see, it's corrected. All right. So, but when you proceed, I think we have another one here. So this is telling us that when you place your cursor on R, it's telling you that it's either A or G. All right. It's either A or G. So, but what's the most expressed here is G, and what's the most expressed here is A. So what you ought to do here right now is you change something, and what you need to change is you impute the one that is most expressed. And here we have G to be most expressed, so you can change this to what? To G. And that's how you keep correcting this this sample. This place we have A to be most expressed, so you can just put that and then it's corrected. And then that's how you keep editing it manually until you finish editing the ITS sequence, because it's not quite too long, and that's why it takes time to actually do this editing. There are other automatic way of doing the editing, which I'm going to show you in the next period or in the next time. So thank you so much. So this is the genomic software tools that you can actually use for different kind of analysis, like I told you. So, and then we're going to send you the software so that you can download it. Thank you. So let me show you the pipeline tools very quickly on the browser. So please give me a minute. Okay, so I'm going to be sharing my screen from here. And I'm going to be sharing my browser. So now this is my browser. Now on my browser, here you can actually see, that I want to search for something on Google. So you can actually do that on your phone or on your laptop as well. So you search for the first pipeline tool, online pipeline tool I'm going to show you for different omics data analysis is Galaxy. Okay, so search for www.galaxy.eu. Okay, www.galaxy.eu. You search www.galaxy.eu. Sorry, let me search that again. Sorry, it's usegalaxy.eu. Sorry, usegalaxy. use Galaxy. Okay, my audio is not clear. Okay, please let me change the laptop I'm using. Hello, can you hear me? Hello, can you hear me? I'm waiting. Okay, thank you. Thank you. Thank you very much. So I'm going to be showing you use galaxy.eu from here. All right. So, and then I'm going to be sharing my screen. Don't forget, we'll send you this the link you will use to download Genius software. All right. Genius software. So let me share my screen from this end. So can you see my screen? We're going to send a link, okay? Kindly exercise patience. We're going to send a link at the end of this workshop. For those that are still on this workshop, we're going to share it, okay, at the chat room. For those, we're not going to share it on the WhatsApp group. We're going to share it on the Zoom, on the Microsoft Teams chat room. So if you leave, you will not be able to access the software. So you have to wait, all right? So you pay the price. Okay, so let's proceed. Let's proceed. So I hope you can see the screen. Use Galaxy, search for use Galaxy.eu. All right. So I'm searching right now. Use Galaxy.eu. Use Galaxy.eu. You can search that on your screen. Use Galaxy.eu. So this is it. This is use Galaxy.eu. Now on this particular pipeline tool, you can do a lot of different kinds of analysis. All right, you can do different kinds of analysis with this particular pipeline tool. So you can do transcriptomics, you can do metagenomics, you can do metabolomics, you can do protein structure prediction, you can and by the time you do that, it will take you to where you are going to register your account on Galaxy.eu. All right, so it's going to take you there to register. Now, the moment you come back to the main page, all right, come back to the main page. Now I'm going to show you certain things or features about this particular online pipeline tools. Okay, so the first one about this pipeline tool is that you can actually use tools. There are a lot of different tools. Now let me just show you that first. Okay, we have the genomic file relation. So we have different kind of tools like convert format, like tools to convert, different kind of format, bioinformatics format, or, genomics data or omics data format. Okay. Then we have the main, genomics analysis, like annotation, like assembly, mapping, variant calling, genome editing, RNA-seq analysis, RNA analysis, epigenetic analysis, phylogenetics, peak calling, different kind of things. So much. We have genomics toolkits. We have metagenomics analysis. So where you can actually use, them to do quite a number of different kind of analysis. So let's look at the genomics aspect. So in the genomics aspect, you can actually annotate different kind of analysis. Okay, you can do different kind of analysis using, this particular annotation. So when you look at PROCA, with PROCA, you can actually annotate prokaryotic genome, annotation, like, bacterial, sample annotation. I hope you can hear me clearly. Can you hear me clearly? So you can do different kind of bioinformatics, analysis. So with use galaxy.eu, and the one I'm telling you right now is PROCA. So I just click this prokaryotic genome annotation, and with prokaryotic genome annotation, you can do quite a number of different kinds of. You can do quite a number of different. Annotation on different kind of bacterial genomes. Okay, on different kind of bacterial genomes. So contigs to annotate. So you need to pick a contig that you want to use to annotate. Okay, you need to pick a contig that you want to use to annotate, and to do that, you have to upload sample. Now, let me just teach you quickly how you upload sample. So to do that, just come here, upload data. But probably before you go there to upload data, you can actually create a history. You can create an history here. So just probably maybe quickly create an history, and just click the plus sign, and an history will be created. And then you can switch to an history as well. Some of the history we created, and you can rename this history as well. So, but let's go to what we are I'm about to show you the other time. So let's proceed. So you can upload sample. So you can choose local files, like samples from local file, maybe genome samples, or different kind of samples that you have. As far as it's NGS data samples, which is next generational sequence samples. So you just click download, and then we have a sample here. All right, we have a sample that we can actually use. So this is a contig. Okay, this is a contig. I think that's a sequence. Okay, so here we have it. We have a contig here. So this whole genome sample of Lactobacillus fermentum, and you open it. So the moment you do that, it will load, and then you start uploading. Okay, you start uploading. Okay, so it's done. All right, it's done. It's done. So close it. As you can see, it's loaded here. So you can, and automatically it picked it. But at the end, if you have a lot of sample, you have to pick them. You have to pick which particular one from clicking here, and then you do that. Okay. Then we have the local stack prefix, which is kind of optional. Okay. Add gene feature CDS, no. So you can just leave it this way. Okay. So when you do, you can just execute. Okay. Once you pick the samples, you could just execute and see what it gives you. Okay. You could just execute. Now we have a number of different kind of annotation. This is just to introduce you to this particular pipeline database. Okay. So the next thing we have here is this could fail because we have not put imputed some kind of files. So which something we're going to do later. I'm just trying to show you some of the things you could do. Okay. So then the other kind of pipeline tools we have is genomic epidemiology. So this is another kind of database. Okay, Center of Genomic Epidemiology. So just search on Google and click it, and then you have this particular pipeline tools as well. So these are some of the pipeline tools we have. Sorry, please give me a minute there. Hello, Sister Shabana. Sorry, please. I think Dr. Shabana is online for aspect on pharmacoinformatics and bioinformatics, a presentation of pharmacoinformatics and bioinformatics. Okay. Sorry, I'm trying to chat up with her to see if she's joined. Okay, so let's proceed. So for you to use this pipeline tools, just click services. Just click services. And when you do that, just click services. And when you do that, you see resistant finder. You see different kind of pipeline tools for different kind of analysis, identification of acquired antibiotic resistance genes. And then you can click res finder to find acquired antibiotic resistant genes. So if you're looking for resistant genes, so just do that. And when you do that, so you can select. It's very simple. Just select the genome that you want to get resistant genes from them. Okay, so just scroll down and pick the genome annotation. So the assembly file, the context. Okay, so just click it and then you upload. So the moment you upload, you'll be able to use this particular pipeline to predict antibiotic resistant genes. Okay, to predict antibiotic resistant genes. Don't forget Center of Genomic Epidemiology. So this is what we have. So this is what we have, okay? This is what we have. This is what we have. So the job is being processed and we have to wait for some time before we get the result. Now, while we are waiting for the result and Dr. Shabana Bibi to join in, permit me to quickly share something with you. So Dr. Shabana is about to join us. So she's going to be presenting to us on pharmacoinformatics and bioinformatics. Okay, pharmacophore and bioinformatics. All right. So I want to show you something quickly from our website. So before the result is done, so just go to our website, www.genomics.com. So we are waiting for Dr. Shabana Bibi to join us. I think she's online, so we are going to let her in very shortly. So, but before then, we want to see the result from this. Oh, said the result failed. The result failed. Can we try it again and see what's the problem? So at times it happens. It happens at times. So let's see. So let's pick order. I think this is what we're supposed to do. So assembly and context. You can also submit reads. Okay. So let's choose file again. Let's choose the file again. And let's upload. So let's go back to our website. So I hope you can hear me. Please, can you hear me? Can you reply the chat room if you can hear me? So these are websites, okay? These are website, genomics.com, Genomics Research Hub. So when you go to our website, okay, quickly go to bio programs, okay? Bio programs. So we have a gift. We have a gift that we want to give everybody for joining our first webinar series. Okay, for joining this first webinar series, we have a gift for everybody, which is bioinformatics workshop and research programs. So, and this particular gift is actually designed to help build your career with us and practically, engage you with research experience. All right, and so that you'll be able to become an expert in your field of study. So, bioinformatics workshops and research program. But we have a gift. All right, we have a gift for everybody who is interested, especially those who want to start, their career as a bioinformatician, all right. So the first thing is, I hope you are there. Go to genomacsup.com bio program, and then so we have in recent time data-driven skills are greatly needed and is shaping the face of different biological related fields such as medicine, okay, agriculture, biotechnology, microbiology. The future of big data is here, where creativity and innovations are integrated with bioinformatics and computational biology to make sustainable solutions in these fields. Genomics tried to provide, okay, on-demand omic data-driven expertise that will enable life scientists to become relevant in the present age. So, and this is how it works, okay? We empower life scientists with in-demand bioinformatics and computational biology expertise and promote competence in the biotech space. Our learning method practically engage students with critical thinking and innovative design to identify and solve biological research problems with real-life omics data, okay? And here we have our workshops, okay? Here we have the first workshop. Now, the first workshop here, like I said, we are going to give you a gift. I hope you are with me. We're going to be giving you a gift. So I think there's a problem with this genome, okay? But I will show you how you can get assembly genome and also use it on Center of Genomic Ethnology. But before we do that, let me just finalize this, okay? Let me just finalize this, okay? So we have get started with fundamentals of bioinformatics. So in case this is your first time seeing bioinformatics, this is your first time hearing about bioinformatics, you can actually get started with this particular fundamentals of bioinformatics, okay? You can get started with these fundamentals of bioinformatics. You can get started with these fundamentals of bioinformatics, okay? So now, these fundamentals of bioinformatics will give you introduction to bioinformatics, teach you different kind of analysis. You'll be able to analyze big data skills, different kind of skills. All right. I mean, big data. Genomics data and NGS data. Do evolutionary study and come up with insights. Okay. Now, the actual fee is $50, but we have a gift for anybody that is interested. Okay. We have a gift for anybody that is interested. So very quickly on the group, if you know you are interested in the fundamentals of bioinformatics and you want to start you want to start your career in bioinformatics, you want to start your career as a bioinformatician, okay? If you want to start your career as a bioinformatician, you want to gain real-life bioinformatics, you want to build your skill, can you just reply at the chat room? Can you just reply at the chat room very quickly? If you are interested to pursue your career, okay, if you are interested to pursue your career, bioinformatics skills, gain bioinformatics, experiences, okay? So can you just signify the chat room? Do you need this course? We want to give you as a gift, okay? Can you reply at the, if you are interested, can you just reply very quickly on the chat room? Are you interested? So I'm waiting. Can you reply at the WhatsApp very quickly? Are you interested? We have, like I said, we have a gift for those that we wait till the end. We have a gift. Yes. Amazing, 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 amazing. All right. So as you can see the price, thank you so much, guys, for your reply. It shows you are interested in this course. It shows you are interested in this course. So if you are, as you can see, the fee is $50, right? And we have a bonus of of $400. Now this $400 is the price for the activation key of Genius software. I hope you can hear me. This is the price of the activation key for Genius software. Okay, this is the price for the activation key of Genius software, which is $400. If you want to buy it now, it's worth $400. But in these fundamentals of bioinformatics, we are going to give you for free. So it means when you get this access to this course, this workshop, okay, it's going to be 1 week workshop. All right, it's going to be 1 week workshop. So you are going to get access to the activation key to activate your Genius software, the first software I show you that can do different kind of analysis. Okay, is there. And the cost, like I said, for this particular workshop is $50, but we are going to give you for $5. Okay, we are going to give you for $5. So if you want it for $5, can you reply with a yes? Do you want this for $5? Can you reply with a yes? You can analyze Sanger sequencing. You can do a lot of editing that we said. And then you have certificates. Yes, you will have certificates, okay? Because Dr. Shabana Bibi is going to be coming for Pharmaco4. And Dr. Shabana Bibi also have a gift. And we will have Mr. Tolu on life science, on career opportunities in life sciences, okay? Career opportunity in life sciences. Dr. Tolu, career opportunity in life sciences. He also has a gift. He also have a gift for everybody. So if you want this for $5, can you reply very quickly at the chat room? Do you want this for $5? Do you want this for $5? I'm waiting. So if you want this for $5, I'm going to send a link. I'm going to send a link for you to join a WhatsApp group. I'm going to send a link for you to join a WhatsApp group. Okay. I'm going to send a link right now for you to join a WhatsApp group. I'm going to send a link. I'm sending a link so for everybody that joined this WhatsApp group, we get this course for $5. It's a gift. It's $50, but we'll give you, and then you get access to this license key for Genius software. You will get access to this, to it. You will get access to it. Join the group. That's the group. Join it very quickly. Very quickly if you want it for $5. If you want fundamentals of bioinformatics, you have not started with bioinformatics before, and you have been looking for where to start from, this is it. So, and in this course, you know how to do ITS sequences, how to do molecular identification of unknown sequences, and how to do phylogenetic tree for whole genome sequences, and do quite a number of different kind of analysis for Sanger sequencing and NGS sample. You get access to pipeline tools as well and certificate of completion. So quickly. So quickly join the WhatsApp group. Quickly join the WhatsApp group. So that's the first gift. We have other gifts that we will show to everybody because we have different kind of workshop, as you can see here. We have comprehensive functional genomics. We have NGS data metagenomics. We have microbial genomics, probiotic microbial genomics. We have pathogenic bacterial genomics. We have variant calling and effect prediction.
We have plant genomics, we have therapeutic peptide design and development workshop, all right? And then for the research program, we have a research program as well, 3-month research program. And for this 3-month research program, we have pharmacophore and bioinformatics research training, okay? Pharmacophore and bioinformatics research training. And Dr. Shabana Bibi has a gift. Dr. Shabana Bibi has a gift for this pharmacophore and bioinformatics research training. She has a gift for those that we wait. She has a gift for those that we wait for a session. And I still have a gift for all these courses. I have a gift for all these courses. And this, for all these courses, like this fundamentals from mathematics, is $80. But I'm going to give you a gift. I'm going to give you at a very, very discount price for you to afford it as quickly as possible. All right? As quickly as possible. But only if you wait. Only if you wait. All right? So I'm going to be calling Dr. Shabana Bibi. I'm going to be, she's going to be coming up right now. Okay. Time is going. For those that want fundamentals of bioinformatics, for those that want fundamentals of bioinformatics, join the group very quickly. Join the group very quickly. And we're going to send a link for you to pay. Only for those that pay, you have between now and tomorrow. For those that we pay now, for fundamentals of bioinformatics, $5, you are going to get access to, I'm going to give you 50 pipeline tools. 50 pipeline tools for different kind of bioinformatics analysis. For those that we pay now and send the payment link to the group. Okay? So I'm going to allow Dr. Shabana Bibi to join. I still have a gift for all these courses. I have the gift for those that will wait for all these courses for metagenomics analysis, functional genomics, microbial genomics, variant calling and effect prediction. Dr. Shabana Bibi? Dr. Shabana Bibi, I believe you can present now. Dr. Shabana. Dr. Shabana Bibi, over to you. Welcome, Dr. Shabana Bibi. Can you unmute? You can unmute yourself now. You're welcome, Dr. Shabana.
Can you see me or can you hear me? Hello.
Yes, we can. We can.
Oh, I'm really very sorry. Actually, I was thinking maybe you cannot see that. Oh, okay.
We can. You can go on now. We can.
Right. Just a moment. Is that my screen is shared to you? Hello.
Yes, yes.
All right. There was the course outline, the pharmacophore and bioinformatics. I think that is shared by Ibrahim brother to everyone. I will just go through the contents of that specific course which we are going to offer you. And we are expected to start it by the start of March. And I think the other things he has already told you in the briefings and different joining program and everything. Exactly what amount and what kind of things he has mentioned to you, you can deal about that things with Ibrahim brother. But I will just simply let you know what actually are learning outcomes we are supposed to go for in pharmacophore and bioinformatics course. We will design a computer-aided drug design and discovery pipeline. I will show you some of my published paper to understand what actually computer-aided drug design and discovery pipeline related to pharmacophore and bioinformatics. That includes specified techniques such as molecular docking, pharmacophore modeling, virtual screening, and admit or pharmacokinetic profile estimations. And for that case, we will use the different ligand data set that can be synthesized or that can be taken from different libraries which are publicly available to us, and we can freely use that. And the molecular target that are also available in different databases. And if in case the target is not available, we are ready to model that specific sequence into structure. And the other ways, the third most probably option is the ligand based or structure based for pharmacophore model. If we have the information of ligands, then probably we will go for the ligand based. Or if we have the protein structure information, then prefer to go for the structure based pharmacophore. There is something coming. What is that? Okay, no. So, critically, we will understand the role of different computational tools, servers, which are useful to identify different drugs and to understand the mechanism of different diseases related to drugs in the specific signaling pathways. In this way, we will be able to design some hit to lead identified compounds, and from that way, we are able to design some kind of pharmacophores that would be the chemical entities which are having some specific activity, and on the basis of that activity, we can go for the structure to activity relationships. And for that case, we have different online freely available tools, we have different licensed tools, and in this specified course we will go for the introduction of computer-aided drug design. And for our specific course, we designed this course in Saturday and Sunday, and the classes would be probably 6 to 8, like 2 hours each day. And after 2 hours there would be a small assignment given to you for the next day, and we will probably discuss that assignment as well. And similarly, in each case, in each week, we have different contents, and that contents already been shared by my brother Ibrahim to everyone. And I am not going to discuss these things, but the thing is, if you people are interested in this specific course, we designed this course for the 6 week, and after 6 week, there would be a project presentation, right? I will teach all techniques, all kind of data applications, all kind of targets retrievals, ligand retrieval, and how, the different application of different computational techniques over that. And then from the raw data to significant information would be retrieved until the 7th week. And then you would be able to present some of your results in front of us, and then on the basis of that we will do some assessment, and on the basis of assessment we will provide you some results in the form of marks and the grade as well. And in the last 8th chapter, like 8th week, I will discuss a kind of a research paper. On the basis of that, we will able to write a research paper. And when we would be able to write a research paper, definitely we will have an opportunity to publish it in different journals, because I am already working as in the editorial board of different journals, and I have already published more than 60 research articles. And you people, if you have any question related to research articles later after your 7 week, you can ask me, and we will use the last month after 8th week for the writing, for polishing, and for publishing of that specific research paper. And actually, these all techniques has been applied in few of my research publications. And I want to share that paper with you so that you may understand what she is already talking to you. There is a paper published, an integrated computational approach for the plant-based protein tyrosine phosphatase non-receptor type 1. In this case, this is my, like during my PhD, I published this paper, and this paper is actually about some drugs which was retrieved from the public data, and they were already showing some kind of potency or some kind of IC50 value, which is already retrieved by some people, and in the different regions of the world, and they are from the source of plants, and they were already studied for protein tyrosine phosphatases. Like we are going towards a targeted like screening approach. That's why in this case we use a set, and on the basis of that in specific set we designed some pharmacophore features. These are the pharmacophore features present in that specific data set. And from that data set, we retrieved how many hydrogen bond regions are important, hydrophobic regions are important, aromatic regions are important, hydrophobic acceptor regions are important, hydrogen bond donor regions are important, like what kind of conformation each compound could possess. And there are the features which are actually important for creating some specific model. And the model fit value, that how many features are actually significant with the specific one model. Usually this kind of diagrams are created by some softwares to generate the specific idea of 3D pharmacophore. And when I will teach these things in course, I will make you able to draw the 2D pharmacophore, 3D pharmacophore. And after this pharmacophore generation, my purpose would be you should know how to generate some models from the specific set of compounds. And from the specific set of compound, you would be having some models on the basis of that specific, overlapping, or you can say shared features, contents or share feature entities or active parts of compounds. We can able to generate such kind of models, and on the basis of that model we can go for the screening. Like further screening would be, in this case I want to show you some of my material and methods. Initially we go for the data set identification, and then we design the pharmacophore model of each compound. And from each compound, we designed a pharmacophore model specifically, which is a shared feature pharmacophore. And then on the basis of that shared feature pharmacophore, we able to select some compounds. And from that compound, we perform docking interactions of that specific ligands with the specific protein, tyrosine phosphatase protein. In that case, when we got that specific important hits, we go for the admit calculations, and then we go for the lead identification. In that case, there was a diagram or a pathway that time we generated. This paper is like when I was I was having a very basic knowledge of these specific techniques, but now we have improved our special things related to pharmacophore, related to docking, related to software, related to different server. So we will improve it. I just want to show it. This is because the simplest form of my publication related for pharmacophore. You can design such kind of paper very easily within 1 month if you go through our specific course, which is 3 months course. 2 months would be the teaching, and 3rd month will go for the writing and polishing of your research paper. And then definitely I would help you for the publication of that specific research paper. In this case, we generate pharmacophore, then we go for the database screening, then we go for the interaction analysis, and then on the basis of that, we go for the drug-like filtrations, and then virtual screening hits identification, and then we can able to produce some specific compounds which are having the most important or most active entities that are responsible for some activities in the specific like protein tyrosine phosphatase or any other targeted pathway. This would depends on you. I will let you know how to select the proteins from the specific targeted pathway and how you can study this kind of interactions between the residues of targeted protein and the atom of different ligands, and what percentage would be acceptable and what kind of these specific interactions are. Interactions have different types, and usually people don't know what type of interactions should be acceptable or what type of interaction should be like ignored and not important for our discussion. So we will discuss these everything in our specific course. And other than that, there is another publication related to molecular docking study and pharmacophore. And in this case, also we have designed like some 166 compounds. Initially, we have selected for the specific target, and then we created the pharmacophore model, and then we go for the zinc database, and we have selected the certain compounds, and on the basis of that screening, that was specified with reference to the pharmacophore model which was selected, and we have screened the virtual hits. That was also important on the basis of that chemical entities which are important for the action of some drugs, and then we screened out some important compound on the basis of interactions, and laterally we suggest these compounds for the in vivo and in vitro validations. But the thing is, like, such kind of publications are important. In that case, there are so many other papers. You can just go and check out pharmacophore based virtual screening. And right now, pharmacophore software is not in my laptop, so right now I am not going to teach you. This is just the introduction. What are the specific things which we are ready to teach you in our specific course. For my introduction, Dr. Ibrahim already told you that I had my PhD and postdoc in bioinformatics from Japan and China. So now the thing is, I will go a brief overview of the specific bioinformatics, computer-aided drug design, or pharmacoinformatics. Pharmacoinformatics is actually the field which is related to the design of different drugs using computational applications. And what is meant by drug? Usually, you people, everybody know there are several organic compounds or chemical entities that are significant for the treatment of some disease, and they have some biological activities or functions, and they are targeting some specific pathways and having some affinity, selectivity, or stability with a specific protein ligand based therapeutic options, and that is a specified drug for your body or for different diseases, and that can be computationally identified because there are several ways by which we can design some drug and compare it with the FDA approved drugs. And hence, if we have such kind of information, which is like you see, there were COVID, and during the COVID-19, there were so many publications. They have the reproposed drugs. Reproposed drugs is that which is already published drugs, already available drugs, that structures are available in different databases. They just go through that drug structures from the database and then apply different computational applications and suggest it for a specific disease, because they already know that specific drugs are already showing some kind of interaction for HIV or some other viral compounds. That's why they are just going to compare the result of that specific COVID-19 protein and the different other viral proteins. On the basis of that, they would be able to suggest that there would be a hypothesis that if they are showing certain kind of values within the range of acceptable specified criteria, as well as the interactions with the specific macromolecular active site residues, so they can repropose the drugs. And in our course, we will teach you how to select the molecule, how to select our drug, and how to prepare that structure for different kind of softwares or servers. And I will teach you molecular docking with the freely available servers as well, or some software, as well as I have some licensed tool like MOE. I can also teach you from that. So the thing is, we will try to get more and more information related to drug and its mechanism inside some specific pathway. And then we will understand why we are requiring such kind of things and why we are using computational methods. Because in the case of drugs, you have two options. Like sometimes some of your drugs are active, or they are enhancing some activities. Sometimes your drugs are like they are inhibiting some activity of the protein. This can only be understandable if you have some information of softwares, you have some information of literature, or you know which protein or which drug actually you are using for your specific study. If you are using wrong information during the collection of your data, then your drug design projects cannot be acceptable or could not be published in a very good impact factor journal. So usually such kind of information we will retrieve and we will teach you out. The drug design and discovery, you people better know, because maybe before starting joining our group, you may have some knowledge of drug design or discovery, and why we are going to design some drugs, and why they have a complementarity with the different shapes or molecular targets which are already available, and how they can interact or bind. So we will study in this specific course about the drug discovery and development pipeline, why initially preclinical studies, how much time was used, how compounds are identified, and now what is the difference with the traditional method, and now the novel method of computer-aided drug design. In traditional method, you was able to design the compound, right? But there were the chance of so many mutations, so many other misleading in the labs. There was several other issues. You have to make a high throughput screening approach, you have to design a big libraries. But nowadays this problem is going to finish by replaced by the available databases such as Zinc or some other databases. You just go to online databases and collect the different compounds, and you can able to design some drug on the basis of some information which is provided as a FDA approved drugs, or sometime it is provided in literature, or sometime you can even design by de novo approaches as well. So you will read about the high throughput screening. I will provide you some notes, some lectures, as well as some books related to this computer-aided drug design. And you will read about the virtual screening by using pharmacophore analysis. Like you will having some data set of drugs, and we will design some pharmacophore, and then we will do virtual screening of large data set, and we will identify virtual drug candidates that would be promising candidates. And the merit of this specific computer-aided drug design or pharmacoinformatic approach is the cost of our identification would be low. We are not supposed to go for the synthesis or non-synthesized compound at the earliest stage identification. So we would be having a chance of more number of compounds that can be virtually identified. And similarly, in my published paper, this protocol is already defined there. How you select a target, what kind of techniques can be applied, then how you will find a hit, how much time it will be used by computational method, and how much this time is used by the other experimental methods, and then you will move to hit to lead identification. Then you can optimize your structure. If your structure activity is not so good and there is a problem in the pharmacophore, and you want to delete some specific entity from your structure and want to design a new compounds, and the basis of that you can go for the structural optimization or analog design. Such kind of things can be also done in the computational drug design, then lead refinement and optimization. These lateral method would lead towards experimental or lab work. But initially, what kind of compounds you can bind, non-covalent, reversible covalent, or irreversible covalent, the knowledge of these drugs I will teach you in the specific course. And the basic modeling strategies, I will let you know what are the drug structures and the receptor structures, and if they are available, you will go for what technique, or you will use what kind of software, and you can retrieve what kind of results from them. So we will try to make maximum learning in this specific course. Different databases, I will teach you. Different software, I'll teach you. Which software I have, I will definitely share with you. And these are some Swiss ADME servers or some other servers which are used to identify the toxicity of say, different drugs. We will try to learn it about each and every specific online server as well. And similarly, there is another one. Then we will move towards a specific technique like molecular docking, protein ligand interaction analysis. We will read about what kind of software and how they predict some drug interactions with a specific protein. We will know about the different algorithms which are actually dealing with the specific docking protocols. And we will learn about how to use the specific executable program. I will teach you a few of the command prompt applications which are linked with some softwares. And other than that, which requirements for the installation I will teach you. Other than that, I will show you how protein interactions. This is the picture of our protein and the ligand. And then we will teach you from which software or from which server you can able to get the protein structure. If these protein structures are not available in these servers or in these specific databases, then probably definitely we will go for the knowledge of homology modeling or some protein prediction from the sequence analysis. We will able to get the sequence from other databases like UniProt, and then we can design or we can model the structure into 3D by using some specific techniques. And then we will explore it in different databases from where we can get the compound, and we will see how the docking in different software works, analysis by different software. Like these are the three different programs showing the interaction analysis. First diagram is from the Discovery Studio 3D presentation. The other one is the Discovery Studio 2D presentation, and the third one is the Lig Plus or Ligplot Plus. So I will teach you maximum of these, and similarly, if I suggest you to read about the structure-based 3D pharmacophore or alternative to docking experiments from different research paper before joining our course, that would be much more better. Like these are the specific pharmacophore databases or different softwares which are available to us, and we can able to learn about it, like Ligand Scout and the docking comparison. We will go through it, and we will see that how our drug is actually binding in the specific active site, and how these entities are showing that these are the specific different entities which are able to present some activity or some specific, like a compound part. Like you can say there would be some hydrogen bond, there would be some hydrophobic areas. That specific area, that specific one specific feature is showing some activity. So such kind of things we will read in the pharmacophore. And these pharmacophore, like I'm not going to teach these things right now, but if somebody is interested, we can go through it. Like we will definitely go through it in our course. This is just to show you like what kind of pharmacophore specified things would be. Like there would be pharmacophore overlapping as well. In that case, we see that the protein ligand complex, and then we create different graphs on the basis of that. We overlap them and we check which specific entities are overlapping to make a specific model. Such kind of things we will study in there. There would be some shared feature pharmacophore. We will see which like set of 10 compounds, 20 compounds, how the compounds have the features which are actually overlapping or sharing some specific features, and showing that these are the features which are responsible for some activity. Similarly, there would be some mutual features as well. Like that is one is shared and the other one is I'm forgetting the word exactly. Merge features. Yeah, in both cases we will study about every type of pharmacophore, and then we will compare what kind of results from the docking and pharmacophore are coming, and specifically we present both kind of result in our papers, and that has been published and getting like approval right now from different publications. And you can read these things before joining, and I will let you know how to perform it with different softwares, and then we will lead towards the publications. This is what. And other thing is, yeah, if you people want to know who I am, you can just go to the website of my university. There is my profile present over there, and there is my research gate. If you want to study about my research publications, then you can even check it out, different publications I have, and right now I'm working on. So if you have interest in specific drug design course, you would able to know most of my papers are related to genomics and drug design, because a little bit information I have from the genomes, and my postdoc is actually from the comparative genomics. That's why I have some publications related to that well. And other than that, my most favorite topic is computer aided drug design. So most of my publication related to that, and I am interested to teach you that. So these are my online profiles. You can check it out, and if you have any questions, you can ask me right now related to specified course. Ibrahim brother, I think you should open or give them access to ask the questions. Hello.
Hello. So if you have a question concerning this,
Brother,
please send it on the group. We answer it right away.
Allow them to ask the question, because I just here to let them know that we are going to design a course. We are offering them by the March, and if somebody is interested for that specific course, we have a 2 months learning, and then third month is related to research publications. Or if somebody have any question related to my specific course, then ask me now.
Yeah, please, if you have a question, you can ask.
But one by one, not everybody collectively start.
Yes. So please, somebody should ask a question, because we are soon going to have
those people who has already shown the interest and registered. If they have any question, they can ask me as well.
Okay, thank you. So because we're going to have Tolu Obatunde to join us. So meanwhile, Dr. Shabana, please, can you allow me to share the course, your own aspects?
Okay. Yeah, you can share. You can share.
So, but meanwhile, is there anybody with question? I don't think there's question, because we asked them to submit their question on the WhatsApp group. Okay.
Ah, really?
Yeah, because this chat room is not working, so they are using the WhatsApp group.
Okay, so okay. If you just let me know their question from the specific chat box, then I can answer them.
Okay. All right. So please, before Mr. Obatunde present on life career opportunities in life sciences. All right. So let me quickly share.
There is actually, this is the era when everybody need the money, right? There are so many freelancing activities on the basis of computer aided drug design. Usually people don't have so many hardwares and softwares and the big analysis and something like that. So if they are inside home and they just have a laptop, so for Maco4 based screening, molecular ducking, admit prediction, these are just on your tips. If you just learn it, you can able to produce a research paper within 1 month. I assure you, if you have the good writing skill, I can let you know you can produce one paper in 1 month. And if you have like 2-3 publications and you want to go for the admissions in PhD or admissions in like postdoc anywhere, so it will help you a lot. Usually in my country, the people don't have their research publication as a first author. They have publications, a lot of publications maybe, but not as a first author, because their contribution is a little bit and somebody gave them name in their papers and so and so. But in this case, you would be able to generate your first author publications. And on the basis of first author publications, you can say you definitely go for any kind of interview, any kind of PhD applications, and you would be selected.
All right. So now for Pharmaco4 research training, all right, on the aspect of Dr. Shabana.
I already taught these courses in different universities as well. I already teach this course a few months before in my university to different faculty members as well. So everybody is keen to learn about such kind of things because this is a very small thing and there are a lot of benefits in sense of freelancing. So if somebody who is dying to do so many experimental work in the lab, and then after that expecting to have a research publication, that may take 1 year or 2 years. But in this case, like if on the basis of computational techniques, you are able to generate one research paper within 1-2 months. If you are really, really desperate or you are motivated to work for it. And we are here to teach you because we already have done this work, and in last year I have more than 40 publications. You can just go to my profile and you can check it out. I'm not like a super special thing person. I am just like you people. I just learned by myself. I just get information from different resources and try myself. But now I am at the stage that I can teach anybody. So if you people have a little bit knowledge about it, you just read about it, what is that and why I am talking about its importance. You just see my different publications. You can see that are already published in Q1 or Q2 publications, and they have the good impact factor as well. So you can also produce it. And for publication or for publication funding or different other things, I'll definitely help you out if you are going to take this course and actively work with me.
Okay. So concerning her, all right. So I'm going to send another link, group link. So if you know you want to participate in pharmacophore and bioinformatics research training for 3 months program with research paper, the fee is $300, but Dr. Shabana Bibi is going to give you 50% discount, which is $150. Okay, 50% discount.
You know, $150 is nothing. Just $50 for 1 month is not a big amount. But when you will learn about everything, you will see that you had paid nothing to me. But I have a keen interest to teach everybody that what I am doing, everybody should know and perform their best. I don't know, but I have this like, what can I say? This is my habit to teach so many students in Pakistan as well. I taught some student free of cost as well, but the thing is this program is launched for specific purpose. That's why we have some short fee to you. So you should pay for it and come to learn with us.
Okay, so this is it. So I'm going to send a link for pharmacophore and bioinformatics research training on the group. In case you are interested, don't forget it's $150. Okay, $150. That's 50% discount of the actual fee, which is $300. So I'm going to send a link.
You are going to pay $150, and $150 even in Pakistan is not a big amount for that specific 3 months course. I have seen usually people have workshop just for 2 days, and they used to ask for more than 50.
All right. So please kindly let's have Dr. Tolu. Mr. Tolu. All right, Mr. Tolu, are you ready, sir? Hello, sir.
I want to know how many people are actually interested in this course.
Yes, I'm going to send a link, the group link now, so that they will join.
I really appreciate if you people go to my specific profile, just read about some of my paper, and then understand what actually you will study in this specific course. It would be more appreciating.
Dr. Tolu, are you here, sir?
Hello.
Yes, sir.
Hello, amazing.
Yes, sir.
Hello, can you hear me at all? Am I?
Yes, I can hear you. Please say.
Oh, amazing. Thank you. Yeah. So good day, everyone. I believe you. It's been an amazing time so far. So
you are interested to take our specific course?
No.
Sorry.
It's presenting. Okay. It's presenting.
what he is presenting. I'm sorry. I was thinking that he is the student going to take the course. Sorry. Please continue.
No problem. Okay, so okay, I'm going to be sharing my screen in a minute. I'm coming.
Sure.
Okay. Good. Coming, give me a moment. Okay. Okay. So I don't know if you can see my screen. So permit me to I'll be sharing from my phone. So, but I believe you can see my screen. Can anybody see my screen?
Yes, we can see you. Continue, please.
Oh, you can see my screen clearly. All right, thank you. Okay, so I'm going to be sharing on, my name is, I'm going to be sharing on career guide and mentorship. Okay, basically streamlined to life sciences, even though I may take it a little bit deeper. So, but it's going to be an exciting time to actually discuss on this particular topic, because it's actually a major topic in anybody's life. Major topic in anybody's life. Okay, so my name is Tolu Abatunde, and I am the CEO and co-founder of Great Plus Africa. And what Great Plus Africa is, it's an edtech startup here in Nigeria, and we have an amazing product that solves problems in educational systems, structure and system for the African populace, majorly for Africans, but our solution is not only based or directed towards African. And so part of one of the things we do is career congruence with personality, and that is going to be one major part of the product I'm going to be exploring in this webinar. Okay. So basically, if you are on this webinar and you are listening to me right now, one of the things you should expect in my session is probably, I don't know, it depends on what category you are right now. Okay, and there has been amazing. There has been amazing sessions before, before myself, before this session, Mr. Luashe Fumi and Doctor has actually shed amazing light on some crucial part of this webinar, and which I believe is going to help you. But now, right now, what one of the things you should expect from this session is finding a best fitted career path. So I believe, just like I mentioned, it depends on the category you are right now. I believe some of you guys are probably in a junction in your life where you're wanting to select a career path for yourself, be it in life sciences or in any other part of the professional space. Okay, so my session will help you do that. We help you have clarity on actually finding the best career path for yourself. And one other things you should also expect is career personality congruence. And this is actually very key. Okay, this is actually very key. Career personality congruence means that you selecting a career that suits your personality. This improves the level of probability of career satisfaction. And this also improves the level of how successful you can be in that line of professional space. Okay, you cannot really be successful if you are not satisfied with the kind or the type of career that you are pursuing. And you cannot really have ultimate joy or fulfillment in a career if that career does not gel with your personality. So, but the problem has been that the ability to be able to understand your personality, because as humans, we grow, okay? And who you were before in 3 years ago is not what you are right now. And so it's most of the time quite difficult to be able to pin yourself to something, and sometimes it's kind of get tricky to say, oh, this is what I think I really want to do with my life. Especially some people who can be like myself. If you're very versatile and you know how to do this, you can do that. And probably when you're still an undergraduate and all of that, you battle with a lot of decisions and all of these things. So it's something that can help if you actually know how to find congruence between your personality and your career and what works for you best. Okay, and then so you should also expect career satisfaction, just like I mentioned, and of course mentorship. So it's quite a lot to within the few minutes I have, but I'll do justice to a crucial part of all of this. I may not be able to kind of begin to list everything that we have, or you can actually walk around probably in life sciences and all of that. Okay, so even though I'm not a principal this in life science space, but as an educator and an edtech startup owner, who has actually done a lot of research into different careers and also personalities and how to help people and students and professionals find the best career path for themselves. We have I can speak on a vast of premises. Okay, but because of time, I may not be able to do that because I really, really want to really touch on the core, the first thing to actually consider when you are when you are selecting the best career path for yourself. So okay, so but probably after now, okay, I'm still going to talk about that. My team and High Grade Plus Africa, we are running. We're going to be running an intensive career mentorship boot camp, virtual career mentorship boot camp, next month and also the month after. So probably in the boot camps we have enough time to actually stretch all of this. But right now, I'm going to actually be talking about how to really the core to actually really consider when you're talking about selection of the best career path for yourself. So, okay, let me give a brief on just a brief bow on Grey Plus. So, just like I said, Grey Plus Africa is an edtech startup, a platform that helps students and professionals find career and personality congruence. That's the connection between personality, who you are, your core person, and the kind of career you actually select and go with. And we do this by helping you explore relevant career paths and discover a good fit, as well as improve your cognitive ability to enhance success in that particular career path. Okay, so we help you journey through selection of your career path and also equip with cognitive skills. That's a mental capacity building courses to help you become the best version of yourself, fulfilling that best fitted career path that you have been able to find. Okay, so and these are the two solutions we actually bring to the market, and which is so much needed. Okay, we are invariably, although this, just like I mentioned, we have done this for students, we have done this for professionals, we have done this for entrepreneurs, we've done this for civil servants and all of that. Quite a number of them. Our product have been able to reach more than 10,000 people. We have personally directly trained more than 600 people to help them through these things. So, and it's something that is key. Yes, because not just Africa, in the entire world, there's a need for people who drive career success more home by actually finding the best career path for themselves. So, okay, so these are the two things we do in Great Plus Africa, and that is that. So, okay, let's dive right into these things. All right, so if you can see my screen, you see I have three things funnel into career path, and then career path funnel into societal interactions. Please pay attention. Okay, pay attention. This is this at this point I need your 100%. I need you 100% to be here. I need your presence 100%. So if you have distractions, you may as well want to get away with them. Okay, do away with them and pay 100% attention to what I'm saying. It will really help you. Okay, so so because not just that you're looking for career path, probably you are ready, even you're already thriving in a particular career or a particular professional space, and you want to really understand how career satisfaction works, how to probably even you want to best analyze yourself in that professional space. You need this. Okay, so three things to that funnels into career path. We have your class category, especially when you're in high school, when you're still learning a lot and you're about to make decision on the particular kind of class category you want to actually journey with. Okay, so particularly in Nigeria here, we have class categories divided into three major parts. Probably you want to go with sciences, you want to go into social sciences and commercial and businesses. So you want to go with art and environmental and societal subjects of courses and all of that, and you can decide on all of these class category. And in the high school is your most crucial decision making time, and in the high school is also your most vulnerable time. I know you agree with me, because in that part of time of your life, you do not know so much about yourself too much. You expect, in fact, as a matter of fact, you are just discovering yourself in bits, in layers, and all of that. So, and that is the time where you are meant to make the most important decision of your life as to what class category you want to journey with, which will eventually determine the career options that will be spread before you. Okay, and all of that. So this point funnels into your eventual career path. And also we have undergraduate course of study. After you have been able to make decision in your class category, when you're now going funnily into a institution of learning, maybe it's university, be it a college of medical sciences, college of engineering, whatever it is, after leaving high school right now. Those times you actually also need to make decisions on what course exactly you want to study, and said that course will also define the career options, okay, that you will settle with in the future. Okay, so and then the third one is, be it when you're in school or you're out of school and you're already in a particular line of professional space or a career path. Okay, you want to make a switch, okay, from one career to another career, probably because of career dissatisfaction or probably funding and all of that, and then you want to make a switch. All of these three things funnels to a solid and concrete decision that turns out to be what kind of career path you're going to actually be settling down with. And your career path also now funnels to your societal interactions. That is how you interact with the society. So your career okay, or your career path in any way, is actually how is that tool, okay, of interaction with your interactions with the society. That is how you deliver your solution to the society, and that is also how the society also remunerates you, rewards you, celebrates you, and all of that. So it's basically the connection with you and societal interaction. So that is very key. Okay, so most of the time, even in the general scope of things, your career really defines you a lot in the society. Okay, sometimes even people tend to define themselves by their career. Like, I am about medical professional, I am about chemist, I am an engineer, I am a doctor of philosophy in this particular part, I am an entrepreneur, and all of that. Your career eventually really defines you a lot, and that is why you don't joke with it. Okay, so that is about that. So okay, so and right now I'm going to give you stats from Africa, which is actually just a tip of iceberg to be able to actually analyze what is really going on in the entire world right now as we speak to you. Okay, stats from Africa shows that more than 60% of youth, okay, I find it difficult, okay, to find the best fitting course of study to select in university. More than 60%. That means it's a huge problem. It's not that they don't eventually settle down with one, but they struggle to really find one that best suits them. Okay, and that's where we have a lot of career dissatisfaction in the society and all of that. So and that is really disheartening. And one of the things that, if you can see my screen, I listed to the major things that are actually causes career dissatisfaction. We have parent influence, we have peer influence, we have most popular courses and all of that. Okay, so the parent influence can be especially during high school, like, oh, I have a friend of mine whose daughter is a medical doctor, and I want you to study. I want you to study medical as a medical doctor. Or I have a friend of mine whose son is a lawyer. I want you to probably go into that line. Or the father may just like a particular career, all of that, and then just imposes it on the child, and regardless of what the child wants or not. And because they think or they feel that the child doesn't know what is best for him or her, and sometimes because probably the prestige of that career path, or the success story and all of that they've heard. Okay, but success actually is not defined by the general, it's not always defined by the general populace. It's rather defined as how much value a person can actually offer in that line of interest and passion. Okay, so parent influence can be a very huge factor. And some of you listening to me here, probably some of you listening to me here might actually be being pushed into this line, a particular line of courses of study. If you're a student, or even if you're already out of school, the kind of career you're doing based on probably what your parents or your dad or your mommy said and all of that. Okay, so which this may be good, but it's not always good enough. As sometimes the decision of the child is not taken into consideration, okay, at all times. So, and also peer influence can come in where somebody, maybe your friend, your friend is going this direction, majority of the classmates are going in this direction, or while you're in school as an undergraduate, people, this particular part of this particular course or career path or career path is trending, and I think this makes a lot of money and all of that. You just jump on it and start doing stuff. That can also actually be an huge part of huge influence on your decision making. And sometimes most popular courses, sometimes some of us don't know enough, like to know enough options available. Okay, so while we are doing quite a number of research work, especially in the Nigerian terrain of courses available and also career paths available in Nigeria alone, myself and my team, we need to find out that we have more than 250 courses alone in the Nigerian university ecosystem. And I was wowed. I was like, seriously, I never thought about that. Okay, because you are not exposed to a lot of them, and you are not so much clear that there is a course called furniture and interior. There is a course called wood and craft. There is all of that. There are quite a number of them in the Nigerian university ecosystem that what I probably before that time I've never heard about them. Okay, so if you are not familiar with a particular thing, you cannot dream of doing something around that path. So there are sometimes that we are streamlined to most popular courses around, and that also informs our decision of career path. Okay, so and these things can lead to career dissatisfaction if they or whatever you decide to go with does not gel with your personality. Your personality is very important. It's your core person. It's your core value. It's your core person, just like I mentioned, and that is what defines who you are and how you enjoy things being done and how you really want to help your society. So if those things are not if that is not in congruency with your decision, you will find yourself always fighting, okay, and always trying to show up while you are trying to make ends meet. Okay, and it's a statistic that's also proven that even undergraduates at undergraduate level and even in high school level or at undergraduate level and even out of school, in probably in your day-to-day activity, delivering your duty as a professional, your success rate is dependent on the level of satisfaction you derive from whatever you're doing. So the more you love what you do, the higher the probability or the possibility of you doing better and doing much more successful, being much more successful in that direction. So if you don't really love what you are, and it's not just by saying I love it, it's about being. It's actually about things that your personality agreeing with what you're actually doing and all of that. Okay, so. All right. So okay, I'm gonna highlight these things and then jump into something before I round up. Okay, and that is obviously they're listening to me right now. You are either in a point in your life where you want to select a career path, you want to settle down with something, you're asking questions. I've studied in school, or I'm studying right now, or I don't know if there's anybody who has not who's still out who's still yet to be an undergraduate. Probably you're still in high school, I don't know. So probably you're just wanting to select a new career path for yourself. You probably some of you may be listening to me and you are in this category. Some people probably you are already in school and studying, and then you have multiple options right in front of you. Like you heard about this, and you want to really settle for one. And because whatever you settle for will determine what you learn and your drive. Okay, what you really want to learn and what you want to explore and all of that. Okay, so probably you are already thriving in a particular career, and you really want to, but you are dissatisfied. You really feel disconnected. Okay, with the career, even though you are already working at it, but you are not really connected with that career path, and you feel you are not doing enough. You feel you are not optimal in that particular space, professional space, and you want to pivot. You want to switch from one career to another career. Probably you're there as well, or probably you are thriving and doing well in a particular career path, and you are enjoying what you're doing. Okay, but you just want a second career. And what do I mean by second career? Probably you are doing something, you are doing great at this career, building your career, but probably with that, in the jurisdiction of that career, you need to make more money to be able to sustain yourself. Probably that particular career doesn't afford you the luxury to actually make enough to sustain yourself and the family, and just want to do, probably add more. You feel you can do more, you have space and bandwidth for more, and you want to add one or two things to your portfolio so that you can be better and all of that, and you want to do something that will still connect with yourself. So probably you're also looking for a second career, and that is fine. Okay, that is fine. So, and let's take it the deep dive into something more deep. All right. So now you have to know this. On my screen right now, I have career is a means to an end. So just like I mentioned when I started my session, I said I may not be able to begin to tell you all the possible opportunities in life sciences and all of that right now, because I want us to first of all address what you need to be able to make accurate decisions. Because if I just begin to flaunt before you the various type of opportunity, okay, probably we may still talk about that down the line in this webinar or in the boot camp. Yeah, we're obviously going to talk to so much deep in the boot camp, in the career mentorship boot camp later. That's for people who are interested, but I'm particular about you knowing what to do so that you can make a correct decision, first of all. Okay, so, and it's very important to know that career is a means to an end. That is, your career is a tool to achieve something. Whatever career you are pursuing, or whatever career you select, is actually a tool to fulfill something. For instance, if I want to roof a house, and I built the house, or there's a construction going on and the house has gotten to a roofing level and I want to roof it. Obviously, when the purpose is clear, which is roofing of the house, you are all 100% clear on who to invite to do the job. So, okay, so let's say you want to use the iron roofing system. Okay, the metal roofing system and all of that. So obviously that you need engineers, trusses engineers, okay, to come and fix the roof. As simple as that. And the engineers, the trusses engineer, they come with specific kind of tools to achieve that purpose. Okay, so the selection of accurate tools is informed by the clear and the accuracy of the purpose. Okay, so when the purpose is clear, the tools with which you will fulfill that purpose is almost 100% certain that you are not going to make wrong decisions. Okay, so that means I cannot accurately decide on what or which career to run with if I'm not clear on the purpose or on my purpose. So your career is actually a means to an end. Your career is your tool to fulfill your purpose. As simple as that. I'm going to repeat that. Your career is a tool to fulfill your purpose. All right. So it's very important. Very, very important. For instance, let me use this scenario. There's a scenario I used to, because I'm a career counselor, I help to a number of folks and all of that to navigate this part of their life. So there's this popular scenario I use. Let's say you at some point went to visit your granny who lives in the countryside, countryside, rural life and all of that. And one day you were sleeping in your room and your grandma wants you to do something for her before she wants to tell you what to do for her while she goes to the market. Okay, but she doesn't want to disturb your sleep, and then she decides to put some stuff beside your bed, so when you wake up you can see them and go ahead and probably do whatever she wants you to help her do. So when she left and she put stuff beside your bed and you woke up, and when looking at your bedside, you saw fishing nets, you saw a fishing rod, you saw the bait box and all of that. You saw quite a number of stuff like that. You saw fishing line and all of that. You saw the hook and the bait, and so it's so clear when you wake up. You can, by seeing those things, decipher by the operation of the few things around to understand the purpose, okay, or that she wants you to fulfill. And when you are clear about the purpose, you can now make informed decision on how you want to use that few instruments that she left beside the bed, okay, to fulfill the purpose. I'm going to explain that further in a few moments. Okay. So if you look at my screen, you see purpose comes before career. Purpose comes before career. Okay. So your purpose is first to be determined before you begin to talk about your career. That is the best route. Okay. Why you can actually decide on the career you're going to pursue without really finding out your purpose, which is fine. Okay. But it's just going to make it a longer route to take. But the best route to making quick decision on career path, and the best route to making a lot of success, having a lot of success stories in your career path, is to really find your purpose first. And when you see the options, you can really pick the right option for yourself. It's very simple when you really know the purpose. Okay, your purpose comes before your career. All right. So now look at my screen. Look at your screen. You see your purpose is the reason. I defined the purpose here. Your purpose is a reason why or for which something exists. Okay, the reason for the existence of something, and also, or you can regard it as a goal set before someone to achieve in a lifetime. So that goal that that is before you to be achieved. Or you can also talk about the purpose as a cause to which you are driven, or that thing that fuels your passion. So purpose is more like that a particular cause that you want to fulfill, that you are driven towards, and when you know that when something that fuels your passion, something that makes you wake up and just want to get things done and all of that, that is what we call your purpose. A purpose is exactly that thing that fuels your passion. And do not forget, purpose comes before your career. All right. So the question is now going to be that what's the process to distance? So okay, just stay with me. All right. So if you look at the screen, you will see that your purpose funnels to success. When you find your purpose and you are able to fulfill it, remember, with the right kind of tool, which one of them is career, because we have a lot of tools to actually fulfill your purpose. We have resources, we have network of people, we have relationship, just like I mentioned, network of people. We have quite a number of stuff to fulfill your purpose. Okay, that is to achieve your goal in life. Okay, something that drives to fulfill your passion in life and all of that. So, and when you are able to fulfill that purpose, it gives you success. And success, I define that's into three phases. You have impact. Your success is your level of impact, your influence over the next generation or even your generation, and the legacy you build afterwards, and also how much inspiration you can be to the next generation or to people around you. So success is guaranteed within the ambience of how much fulfillment you brought to your purpose. Okay, so and how does it work? Okay, so the first thing is to find purpose, and after finding purpose, you find your career, and after finding career, we have something called career building. You build your career, and after career building, that helps you to double down on fulfilling purpose, which eventually brings you to success. Okay, so successful people are purposeful people. Know that. That's so important. Anyone you see that is successful in any area of life whatsoever, he or she is a very purposeful person. Someone who has a goal. You don't just wake up for nothing. You have a goal in your mind to fulfill, okay, to make the world a better place. Something, your contribution, your legacy, what you want people to know you for, and all of that really helps a lot. Okay, so and so the point is just how do we find purpose and how do we find career together with it? And just like the story I said initially, your grandma dropped some stuff beside your bed, and then waking up you found out that, oh, by looking at these things she dropped beside my bed, she wants me to go to fish. Okay, so the purpose is to go and fish. The tools there, the instrument rather, if I want to use this so that I'll be consistent with my choice of word. The instrument rather are more of like your crew, your few things I'm going to be talking about soon. Okay, like I don't want to run ahead of myself, but the instruments are something that funnels to the pathway or to the process with which you're going to use the instrument to fulfill your purpose. Okay, so and that is your career path. Your career path is a pathway to actually engaging the instrument you have to fulfill your purpose. Okay, so now, yeah, this is something that you need to really pay attention to right now. Really need to pay. If you can actually pay attention to this, you can actually guess anything about the best career path for yourself, even without me guiding you through them. Okay, so so even though like I said, I won't be showing you a list of all the possible careers in life sciences, but I want you to be able to when you see the list eventually, okay, you can really pinpoint that which you really want to be, because when the purpose is clear, the career comes running to you itself. Okay, so the first thing is what you see right now is a diagram, a Venn diagram called the Ikigai model. Is popular with the Japanese. Ikigai is another word, is a translation, or is the Japanese translation of the reason for being, or purpose, just purpose, right? So finding purpose and picking up tools to fulfill purpose, okay, which is career path. So if you look at that screen, I would have really, because this is a talk, I would have really loved to be able to have feedbacks, but I want to really believe that you are listening to me. Okay, I don't know if I can confirm that. Like probably if I can have a feedback that yes, sir, I'm listening, or probably a chat in the WhatsApp chat, all of that. Okay, normally if we had a way, we could actually have this as a conversation, but it's still cool. But can anybody just give me feedback that, oh, I'm listening, I'm following, I love that. Okay, well, I'm going to continue anyways, but I want, if I get a feedback, it will really be cool. Okay, so so I'm just going to discuss this briefly. Okay, and then yeah, that'll be all for my end. So the first thing to know is that you are a purposeful being. You're a being of purpose, and your purpose comes before your career. And when you are able to define your purpose, you'll find your career quickly, easily. Okay, because your career is the tool to fulfill your purpose. And within the career, we now have subsets of tools, okay, that helps you to engage the path way to which you fulfill your purpose. That is, your career is also is an embodiment of some other tools. Just like I mentioned in that story, those things, the fishing rod, the fishing net, they are not really your career per se. They are the tools that forms into a career that helps you to fulfill the purpose. So the purpose is to go to fish, but the career is fishing, right? And the tools for you to fish are the fishing rod and all of that. Okay, so the career is also an embodiment of other instruments, other tools. Okay, and those tools are what I'm going to be sharing with you in this Venn diagram. So if you are able to find out your own peculiar, unique tools, you can easily describe yourself and also understand the best career path that you should go with. Okay, and as a matter of fact, a career path is not something like it's not rocket science, and it's not black and white. That if I don't do this with my life, I cannot do any other stuff. Okay, what a career path can always change. That's why we can have people switching from one career to another by getting to select a second career and all of that. Career can always change, but the purpose remains the same. Okay, so the first thing I'm going to talk about here is, you see on your screen, you see what you love. Okay, that's a very, very pivotal tool that informs your career, which eventually funnels to your purpose. So what are those things you love to do? So in my own words, I can call that your gift and your talent. So what you love to do is what you find easy to do, what you enjoy doing, right? Backdating even to as much as when you are still a kid. Okay, so sometimes to be able to do this analysis of yourself, you may have to sit down and think through back to the years where you were still young. Like, what are those things that really get my attention? What are those things that I really love to do? What are those things that I enjoy doing? Okay, those things, let's say for some people, they love to cook. Okay, some people probably, let me use an example of a boy. Let's say this boy is called Henry. Henry loves to cook. Henry loves to take care of people, care for people. Okay, Henry loves to sing. Those are something that when you say love, that means you can stick to them for hours without getting bored. Okay, and yes, you can stick to them. You can do it for hours without getting bored. You enjoy doing them. Okay, it can be anything. And when you go to the next circle, you see what you are good at. So what you are good at is more of your skills, not necessarily what you love to do. Sometimes some of the things you love to do become what you're good at because probably you went further to learn about them. Okay, so but what you're good at are your skills, something you have acquired by learning, either directly or indirectly. So and let's say Henry is good, out of being being a good singer. I mean, being someone who loves to sing, who loves to cook, and who loves to take care of people, Henry becomes good at cooking. Okay, and becomes good at let's say okay, becomes good at coming, becomes good at something and all of that, learns that skill. The skill is what you learn over time, and the bridge between your skill and your gifted talent is your passion. Okay, what you're passionate about, that's what informs what you're passionate about, right? That's what informs what you're passionate about. And then what you're good at, when you begin to itemize what you're good at, which is those things you have learned, the skills you've learned, or what you have found yourself to know how to do. Okay, out of those things you're good at, what are those things that you can be paid for? Okay, that is what we term as your possible professions. or what you can, yes, possible professions. All right. Okay. And when you also move to the other side, what those things you love to do, out of them, what are the things that the world needs? That is things, problems in the world that what you love to do can solve. And that forms your mission. And also what you need, what you can be paid for, out of those things that you can be paid for, that the world needs, forms your vocation. So the combination of your passion, your mission, your profession, and your vocation is what, in one sentence or in two sentences or in few words, defines your purpose. So let's say Henry knows how to cook, loves how to cook, loves to sing, loves to take care of people, and then Henry becomes good at cooking, becomes a chef, and also learns how to give it becomes a caregiver, and that's a passion, that becomes a passion, his passion, cooking, cooking, and also giving care. And then they could pay him for cooking, they could pay him for medical services, and then what the world needs that connect with what he loves to do is let's say the world needs healthy feeding. And then yes, the world need healthy feeding. The world needs to become a better healthy place. So a combination of all of these things already forms what the life of Henry is all about, which is Henry is all about helping making the world a more healthier place, or yeah, a more healthier place, and a more healthy place about feeding, all of that, and that can actually shape the design of Henry's life, and brings him to a place where he can accurately decide on what career path he actually goes with. Okay. So, and this is very key. This is very, very key when you're talking about career path finding. Okay. So, okay, so oh sorry. So the purpose, just like I want to reiterate now, the purpose is your reason for existence, and is the goal set before you to achieve in a lifetime, or a cause to which you are driven, and that thing that fuels your passion, Okay, so like for the time being, I may not be able to dwell on the list of all of that, but one thing that you've been able to take out of this as a roundup is that your purpose precedes your career, and if you can find your purpose, you can actually accurately decide on what career to go with. Okay, so I believe that that is enough for you to actually stick with for now. So I believe I think that will be what I will go to post this evening from Nigeria. Yeah, I don't know what time you guys are where you are. So we're gonna I'll be giving opportunity for people, for especially this participant of this webinar, if you want to take things further, like you want to I believe I believe I am still very audible. Yeah, let me see. Okay. I believe I'm still very audible. I'm coming, give me a moment. So I believe I'm still very audible. And so I'm going to be giving you guys the chance to actually take a bit more deep dive into this, because obviously I can't talk about everything within the few times I have here, right? Actually, so, and I could only drive home the points of actually coming to terms with your purpose. Okay, so I'll be giving room for people, especially participants of this webinar, to actually, if you want to take a deep dive into career mentorship, like you want to be more acquainted with who you are, you want to really be able to double down, or you need guidance and mentorship. So Great Plus Africa we're going to be having a boot camp, a virtual boot camp for people like you, and we're going to be giving the discount for the participant of this group, of this webinar rather. And I'm also going to be pushing this out like more of like a free gift, even though it's not really free, but it's free because it's going to be discounted for you because you're participating in this webinar. One of the other things we do in Great Plus Africa is to help people improve their cognitives, and we have these amazing resources, okay, of video courses for how you can read and retain what you read, either you're a student or not. Okay, memory, that's what is part of cognitive skills, ability to be able to read, retain, and comprehend faster. Okay, another, and it's a package of two of our courses, the memory part and also the meta attention, ability to be able to pay attention to improve your lifespan. I mean, attention span rather, attention span. Okay, so ability to be able to increase your attention span, because attention span is a big issue in our generation right now because of so many distractions. So these two courses, I'm going to be giving it out for the participant of this webinar for almost free at a very discounted rate. So if you're interested in both of them, interested in getting access to these courses, okay, and also you're interested in the virtual bootcamp, okay, where we double down, talk about career, and we are much more practical, and we take things I'm going to be myself and my team and some other instructors. We're going to take it a little bit more personal, more practical, individually, breaking down, sharing with you our resources, our list of courses, and all of that to help you, guide you through the best career path that will help that will give you satisfaction and give you the most probable successful rate as you journey into the future. You can also Mr. Sheyi will be sharing with you, with the group, because I believe the chat room here is not working just like I learned. So I'll be sending a link to him. It's a Telegram page. So we'll forward it to the group for you. If you're interested, you join the Telegram page. You're interested in the courses, the video courses, the cognitive skill courses, and you're also interested more to actually know more about career mentorship, and you want to explore, you want a more practical, personal exploration of the career path that are available and all of that at very discounted rates. You can join the Telegram page of our class extension, Great Plus class extension, where our facility manager will be waiting to share the two opportunities with you to make your decisions and all of that. So okay, so that's going to be from my end. So I don't know if I have question, Mr. Sheyi. Hello.
Yes, hello. Is anybody with question? Anybody with question? You can drop it at the WhatsApp group if you have a question for him. Meanwhile, we are going to drop the link for you to get access to the masterclass on all that Mr. Tolu have said. I don't know why Dr. Tolu keep coming to my mouth.
That is amazing. Maybe it's something I should explore soon.
So please, I'm going to share it. So is there anybody with question? I hope there's anybody with question. You can. All right. So if there's nobody question, okay. So we are going to round it up for the day. Thank you so much, guys, for joining us. So we'll continue this webinar series next week. Next month ending for the next series, and we promise that it's going to be much more impactful than this present time. Okay. So thank you so much, guys, for joining. We appreciate you. I will send the career guide and mentorship link as well for the bootcamp right away on the WhatsApp group. So thank you, guys. We appreciate you. Thank you so much, Mr. Tolu, for joining us. We really appreciate your presence. Thank you. And we appreciate Great Plus for this partnership with you. Thank you so much.
Thank you. It's a pleasure, and it's something we really appreciate to be jumping on this webinar with you. Thank you, everybody, for listening. And take care.
Thank you so much, Mr. Tulu. So I'm going to be sharing the link now, everybody. Thank you so much for joining. We really appreciate.
And by the way, I Mr. Shei, I shared with you the slide for my session in case if there's anybody wants to follow up with that. So you may also want to forward it to the group. Thank you.
Chats.